Drug development is very expensive and very long process. And in pretty much at any stage of that process, we can fail. So how we can learn from those failures.
I learned a lot in every therapeutic domain about how data, science, analytical prediction, and appropriate monitoring of a clinical trial can lead to success, but also the situations under which there are risks that are generating failures.
What do we need to know about clinical trials? What is the purpose?
Clinical trial is an experiment, and as an experiment, it can be successful. You can do everything you can to make it successful, but if in the end there is always going to be a part of a chance of hazard that makes it a turn on the right on the wrong side.
Thanks to the new technologies and data science, machine learning, we can actually be better in predicting and making the risk, like lowering the risk of failure. This is where we bridge the gap between innovation and scientific discovery. This is where technology meets science, a podcast by Appsilon. Each episode brings you insights from industry leaders who are pushing the boundaries of what's possible in life sciences. Enjoy, and if you like the series, click that subscribe button. This series is brought to you by Appsilon, a technology company that empowers pharmaceutical and life science companies to leverage open source technology for faster, data-driven decision-making in regulated environments. Welcome to the very first episode of Where Technology Meets Science by Appsilon. This is where we're going to explore how modern innovation is changing the life sciences landscape. And today we have a very special guest, a senior leader in biometrics and data science for R&D in the pharmaceutical industry. He's worked in the field for over 3 decades in various roles related to drug discovery, development, and approval. He spent a big chunk of his career at Sanofi as a director in different departments, and currently he's working as a consultant helping other companies improve patients' lives through development and transformative drugs. Eric Genevois-Marlin, welcome to our podcast. It's great to have you here.
Thank you, Nat. Thanks.
How are you today?
I'm very well, thank you. I'm very happy to be here with you and share some thoughts about this this domain which I'm passionate about.
Amazing. So we do speak a lot about successes in clinical trials, but because this is drug development is very expensive and very long process, and in pretty much at any stage of that process we can fail. So today I want to actually focus on that part of the spectrum, but not really to speak about failures and beat ourselves up, but that how we can learn from those failures. But before we start speaking about clinical trials, I'd like you to maybe briefly explain your journey, how you went from being a biostatistician to a director of R&D and data science.
Sure. I was trained, as you said, I was trained as a statistician, and I started to work as a clinical statistician, which means supporting a clinical trial and providing all the methodological and analytical input into the trial in order to design it well, and when it's terminated, to analyze the data properly and to gain evidence about the activity of a particular molecule based on that clinical trial in a particular indication. And then gradually I grew up in responsibility up to become the VP of Biostatistics and Programming at Sanofi. And in that capacity, I was able to overview the development of all our molecules from very early to the approval and the marketing of these molecules when they were successful. And of course, I learned a lot in every therapeutic domain about how data, science, analytical prediction, and appropriate monitoring of a clinical trial can lead to success, but also the situations under which there are risks that are generating failures which can be very harmful to the patients, to the health system, and to the company.
And I guess thanks to the new technologies and data science, machine learning, we can actually be better in predicting and making the risk, like lowering the risk of failure. Before we start talking about the examples, could you briefly talk us through what do we need to know about clinical trials? What is the purpose? Where does it fall into a drug development pipeline? And what can go wrong at different stages?
Yeah, it's a very long and very complex process. It's based on biology. It's based on chemistry. It's based on human bodies and properties. Therefore, it's extremely complicated and it's not completely predictable because we probably don't understand much more than 5% of the biology of the human being. Therefore, a clinical trial is an experiment, and as an experiment, it can be successful. You can do everything you can to make it successful, but if in the end there is always going to be a part of chance, of hazard that makes it a turn on the right or on the wrong side. Drug development is starting when we have a molecule, we have proven that it can have some activity in a biological and a human setting, and therefore we are conducting a series of clinical trials from phase 1 to phase 2 to phase 3 in order to learn from the molecules, learn from the molecule in the patients in a particular indications, and understand gradually, is it safe? Is the patient going to tolerate it? What is going to be the best dose to treat and to cure the patient? How long do we need to treat the patient? Who are exactly the patients that need to be treated? And eventually, what is the benefit that the drug is bringing to the patient? What are the risks associated to taking the drug? And is the benefit risk of that molecule beneficial? In which case, more likely the regulatory bodies in the world will approve this molecule to be marketed. Phase 1 is about finding the maximum tolerated dose. Phase 2 is about confirming that there is a proof of activity, a proof of mechanism or proof of action of activity with the molecule, and to start doing a kind of dose ranging, evaluating the activity of the drug at different dose levels and regimens. And phase 3 is to confirm and demonstrate that the drug is safe and efficient.
And are there any patterns in terms of like when we actually fail more? Is it like the further we go into the clinical trial, the lower the risk, or it doesn't really matter?
Yes, it's really the further you go, the lower the risk. At the very beginning, because you know very little. So it's just like flipping a coin. You don't really know what's going to happen. You have absolutely no control. You know nothing. Everything can happen. Then gradually you learn about how the drug behaves in the human body, what is the level of dose that is likely to have some activity. You learn about the competition. You learn about the chemistry. You learn about how the patient tolerates the drug, and gradually, of course, you know more. You adjust your experiment and you adjust your ambition, your objectives to make sure that you are going to target the right patient population for a disease which requires additional therapeutic options, and then gradually you are going to get closer to success. But even at the very end, when you have reached a positive phase 3 trial that you have properly designed, there's still a chance that for some reason, the regulatory agency will not approve your drug. It may be a risk of 10-15% for a variety of reasons relating to quality of the product, quality of the submission dossier, or magnitude of the treatment effect in terms of efficacy, or assessment of some specific risks that a regulatory body would find unacceptable.
Okay. And do you have any examples of when it actually failed at the last phase?
I do, actually. Yeah, we have been through such an example. Actually, we have even a couple of examples. But the one that is more pregnant in my mind is the one where we failed to provide good quality data sets to the FDA. The drug was efficient, the study were positive, but the submission dossier was not well done. We probably lacked quality control. We were not totally compliant with the requirement in terms of data set structure and standards. And when we submitted the dossier to the FDA, we got a refusal to file an RTF, which led to a delay in submission and approval of that molecule by, I believe, 1.5 years, which is huge considering the costs that are related to drug development. And it's very bad for the patients who are waiting for a solution that doesn't come.
if we're speaking about the costs and investments, how much does it actually cost, and how much do companies invest in this particular in clinical trials, or maybe if you can say it broader, in drug development?
These are huge numbers. I think I would say for drug discovery to approval. So from the very beginning to the very end, we are talking about something around $2 billion for one particular molecule, and it's something that lasts around 10 to 12 years. Of course, there are exceptions. You can go faster. You follow a track that has been already followed by others before you, so you would save time. But in average, this is what we are talking about: 2 billion, 10 years.
And then, so we have direct costs related to investments. So obviously, if our drug gets rejected and we can't market it, then we invested that money and it goes to nothing. But I can imagine there's also some hidden cost related to maybe planning to the whole production line, or maybe thinking about marketing the product before the clinical trial finishes. So what are different costs related to it?
I'm not totally sure about marketing and production. Mm-hmm. Everything that is being done prior to approval is usually counted and factored in the general cost. What is also factored in, which is important, is the cost of all the development of the molecules that will not reach the end. All the drugs that we are developing, for which we are doing experiments in phase 1 and then we stop, or phase 1, phase 2 and then we stop, are also factored in, because this is also what the money that we are spending for what to for one success needs to take into account all the money that is being spent on all the failures, those that do not come to the end, and that's what makes the average so large.
Mm-hmm. And what are the main causes, the roots for failing in clinical trials?
Well, there are several sources of cost of causes of failure. As I said, it's biology, so there's something that we don't know. First of all, you need to design your study properly. So you need to be of extreme scientific rigor. You need to really study very, very thoroughly all the data that exist around there, and that can help you inform the behavior of patients who are taking a standard of care.
And I just want to have a follow-up question here. Like, and what kind of data do you deal with? Is it like, one thing, what are the sources of the data? And also what type of data? Is it like about patients' lives, the drug itself, the molecules, how they behave?
It's all the data everything that we know that we have produced ourselves regarding the behavior of the drug. So how it's constructed, its behavior in the human body, the pharmacokinetic, pharmacodynamic, all those elements are produced by our internal experiments. But you also need to know based on external data, that can be external clinical trials, external registries, external databases with real-world evidence, where you actually can predict and observe and estimate what the patients in your target indication are normally doing, how they normally behave, and how they evolve over time when they don't receive your drug. And then you can build your clinical study taking into account what you are going to compare to, which is patients treated by the standard of care or placebo or particular already approved drugs. And all those elements, you have to accumulate this data very quickly and analyze them very thoroughly in order to make the best estimate and not be too optimistic, and always understand that there are hidden risks, and therefore excess of optimism is a risk to fail the study. But that's one element, that's one cause. Another potential cause of failure is inappropriate implementation of the clinical trial. You go in the wrong places. You don't find the patients because your protocol requires some inclusion or exclusion criteria that are not realistic. So you need to understand what is the real life, what is the life of a patient in this particular indication. So you need to have the input from the patients. The voice of the patient has not been well factored into account in the past, and now we start to listen to them to know what would be a good clinical trial for them. You need to have good quality data. You need to have a good monitoring of what is happening all throughout the story, and you need to analyze the data properly, of course.
Mm-hmm. And has anything changed in terms of the root causes, like when you started working in the industry and then how it is now? Has the technology maybe helped in some of the areas, or maybe it's changed because the way we work changed in the particular process?
Yeah, many things have changed, but I would say 30 years ago, when we were designing a clinical study, we were looking at some publications, which we knew about because we didn't necessarily have the full knowledge, but we didn't have internet. So we had to figure what would be the right paper that could document a little bit the context into which we were going to run our own clinical trials and make our estimate and design our study on that basis. Because we didn't have access to data. The use and the access of data, of massive data, did not exist. I think also in over the past decades, the regulation has been more strict. The FDA has continuously published new regulations. So did EMA and PMDA in Japan and in other countries. And also many new medications have come on the market, raising the bar in specific indications where if you want to add a new molecule in a space which is already kind of saturated in terms of drugs, you have to demonstrate an overwhelming efficacy in order to be worthwhile and to be considered for a success and for approval. So that, of course, has changed the focus to more rare diseases or to rare cancer types or areas where there is basically no therapeutic options. And of course, that makes things way more complicated because these are diseases that are not as well known as can be obesity or diabetes.
Mm-hmm. And this might be a little bit controversial, but how do you see the regulatory requirements that changed right now? Obviously, they're more strict, which comes in favor for the patients because we're not going to release a drug that is not properly tested and verified. But at the same time, do you see that sometimes maybe it's working not in your advantage or patient's advantage because it takes longer and it's more difficult to release a new drug? So essentially, we waiting so long to develop drugs for cancer, for drugs for very, very rare diseases. So, like, what will be your take on this?
My take on this is, first of all, what is predominantly important is the safety of the patient. So any measure that is taken in order to protect the patient is indispensable. And I would say in the past decades, we haven't seen many situations where there have been a kind of a drama in terms of clinical trial safety issue, and that's a good sign, and that has to continue. So I'm supportive of any measures that support the safety of the patient. Second, I think I'm thinking about the FDA in particular because their regulations is usually coming first. They are extremely rigorous, scientifically rigorous, consistent, and they try to maintain that degree of consistency despite all the temptation that can come from the industry to find some shortcomings or some corner cutting in some instances. And I also appreciate the fact that the rules exist and that it's strict and that scientific rigor is of the utmost importance. Now, I think that the FDA is also realizing that the time it takes, the money it takes to develop a drug in situations where there are some urgent needs for the patients to find new solutions, they realize that the paradigm that as it exists today is reaching its limit. It's going to be hardly sustainable for the industry to continue at that space with such cost and such complexity. Therefore, I think the FDA itself is trying to work on alternative ways, such as using more real-world evidence components in a registration dossier, or even using more artificial intelligence, using more modeling and predictions in order to reduce the number of experiments in the human being, not eliminate it, but just reduce the volume that is needed in order to reach approval. I think this work is in progress. It's necessary, but as everything, it takes time, and I think the time we will see the benefit of the modernization and the simplification of the regulations, it will take another few years.
Okay. Let's go back to the examples. Can you mention more of examples from your own personal experience or maybe from the industry that because there was a mistake or there was maybe too much optimism on the way.
Yeah.
The clinical trials wasn't successful.
Yeah, I of course, over so many years of working drug development, I've seen quite a lot and several situations, such as the ones you mentioned. I have one particular example of a development that we did in the cardiovascular setting, where we did a very nice and quite large phase 2 study trying to evaluate the dose response on a particular clinical endpoint. And based on one particular finding, we engaged into a very large trial in phase 3, really large and very, very costly, and this turned out to be negative. There was some efficacy, but not sufficient to reach what we call statistical significance. Therefore, the story was over. And we've had a hard time to understand why we failed.
And when you just want to jump here, like when you say the story was over, so that means like all your effort was...
Yeah. We decided there was no other decision than to discontinue the development of that molecule, because that would have needed to reinitiate a number of very large experiments with the chance of success, which was unknown, but probably the chance of failure was too high. And also it was in a therapeutic domain, in an indication where there were already existing therapeutic options for the patients. I think the error, or at least the cause for that failure, is probably related to the way we have designed the study, to the way we have probably overestimated what was going to be the effect size of the study. Considering that if we had targeted for a smaller effect size, it might have been clinically irrelevant and would have even increased further the size of the clinical trial. So maybe the best option would have been not to go. We decided to go. We failed, and we paid it the full price.
And any other lessons learned from this case?
Well, the lesson learned from this case is that the design stage is, of course, of the utmost importance. You need to do way more research, way more prediction. You don't want to rush to start a study before you have the best understanding of what is most likely to happen. And in particular, the risk of failing has been properly quantified. And I think this is something that we failed to do right.
So there's a lot of guessing at the very beginning. So
there's a lot of guessing. There's a lot of intuition, of medical judgment, of statistical considerations. But sometimes the story is not completely well aligned.
Any other examples?
I can think about a couple of other examples. One was a trial for which we outsourced the full study to a CRO, but I would say it's almost an irrelevant comment. And we did not organize properly what we now call data surveillance, which means while the study is ongoing, you look on a regular basis at what you have collected so far. It's a blinded study, so you don't know treatment assignment of the patient, but you have a kind of overview of what is the average, what is the distribution of your data on the various parameters that you are collecting. And we failed to observe that the key parameter for the study, what we call the primary endpoint, was not behaving the way it had been designed in the statistical hypothesis. In particular, the variability was way higher than what was expected. And there were also another few things that were happening that we did not anticipate, like the number of patients who would discontinue the trial prematurely and so on and so forth. And when the study was over, we unblinded, we found the results. The study was negative for that reason. And this is something that we could and we should partially have detected earlier. We didn't, and as a consequence of that failure, we implemented in the company at that time new processes to review the data while the study was ongoing in order to avoid that a story like this would repeat itself.
Okay, so do you have any more, maybe a few more examples? Anything else that comes to your mind?
I have another one. So for the previous one, I think the key point is to really use every tool that can exist to have a real-time review of what the data that you're collecting are. It's extremely important. and because you really can fail a study for that reason. The last example I would think of is an example in psychiatry, where we did have a positive study. The study yielded a statistically significant difference between our drug and I think it was a placebo, and yet we didn't submit a dossier, or we submitted a dossier and then withdrew it. The reason was that even when you have statistical significance, you still can show that your drug is bringing a modest benefit to what is already existing out there. And this is something that we had also inappropriately designed. We probably set the bar too low. We reached that bar, and when we decided what are we going to do with that, we found out that the drug was not going to bring anything to the patient, albeit maybe some safety concern, some tolerance issue. So we tried to find a way to build a story in which the various pieces of benefit that the drug was bringing could be put together as a kind of a package of evidence that it was going to help the patients, but we didn't find anything convincing enough, and therefore we decided to stop, and that drug was not developed further, was not marketed and not developed further.
And do you have any maybe data regarding like how much of the clinical drugs that go into clinical trials succeed, and what was the percentage that fail?
Those numbers are regularly published. I think that in phase 3, which is really what matters most, because this is where the big cost is, the failure rate for phase 3 studies, I believe, is something around 40%. It's quite heterogeneous between therapeutic domains. It's a little bit higher in oncology, for example, but it's 40% chance of failing in the clinical trial. It's big in phase 3. Yeah, okay. It's big.
It is.
It's huge, actually. It's huge.
Okay, what are the things you also already mentioned that designing a clinical trial is an essential point, like just basically building this strong foundation for the clinical trial is the most important thing. But are there any other elements that you would do differently, having the knowledge you have now from all of the different cases?
Yeah,
both successes and failures.
Yeah. So the design part for me is absolutely essential, and the fact that we have access to more data, to more technology, that we have access to more sophisticated analytical method to build models and prediction, and to associate various components of knowledge on the drug, on the behavior of the drug, on the disease, on the patient, and so on, can really help narrow the space of uncertainty before you start your clinical trial. So that's absolutely key, essential, and we keep learning about that. And the more data we have, the more the technology helps, the more powerful are the computers, and so on and so forth, the more we learn. But we are never going to predict for sure precisely what is going to happen. I don't think it's possible, because this is biology and we don't know it completely yet. So that's one element. The second, I think, which is absolutely critical as well, is how you operationalize your clinical study. You need to enroll patients in your studies that are representative of the real world, and they should not be biased. The distribution should not be skewed because you have introduced some specific selection criteria, which may eventually make your study positive, but may not be representative of the patients who are subsequently going to receive that drug and be treated with it. So have the right degree of diversity in your clinical trials, have the right input of the patients for the clinical trial to be simple, accessible, and I would say pleasant to participate in for the patients and for the investigators, which means the physician, the treating physician, or the hospital. I think also that we learn from using more technology, like digital devices that can collect the data more precisely at the source and avoid various handover of data points from one computer, from one system to the other. I think there are a lot of changes that are happening in the operational space in order to accelerate it, increase the quality, and increase the comfort for the patients and for the nurses and for the treating physicians. I think this is essential.
Okay. Thank you so much, Eric. I'll have two more final questions. One would be regarding the trends and the biggest changes that are coming up in the next maybe 2 to 3 years in the clinical trials area? What do you think the future is going to bring?
Yeah, I think I've mentioned maybe the 2 points which I see are more likely to be predominant in the near future. One is we are going to continue to see more and more approaches around modeling and simulation to test some hypotheses that would normally in the past would have been treated through clinical trials. I think you start seeing some indications for which the label approved by the regulatory bodies incorporate some elements which are the result of a model and a prediction rather than the result of a clinical trial. So that's a very important element. The second one, I think, back to patient centricity in clinical trials, is to have the ability to go to the patient rather than have the patient go to the investigator side. The use of digital devices to collect and to measure some functioning elements in the patients, which are usually very subjectively assessed by one or two particular raters. Now you may have some systems who can give you much more objective, accurate, and real-time data. And I think this is going to transform, accelerate, and hopefully eventually reduce the cost of clinical development, but more importantly, increase the chance of a good drug being approved and marketed.
Okay. And the last question that we want to ask to all of our guests in the podcast. So what is the problem in your field that you're surprised that hasn't been solved yet?
I've mentioned it earlier. It's a good surprise in a sense, and it's less and less a surprise. But the fact that there are not dramatic situations occurring regarding safety of the patient. I think the regulation and the proper care that is being taken by the various sponsors in order to put a drug on the market, they are done in such a way that patients are protected. We are injecting them chemical substances, and there's always a risk. I think that so far, we can see that the clinical trials are done in such a way that they do protect the patient. And there have been a few situations of tragic incidents in phase 1, maybe 10, 12 years ago, with a couple of deaths, which were dramatic in the setting of a phase 1 study. This has not happened since then because I think the right protection have been put in place. And I'm not surprised, but reassured that it continues to be the case.
Okay. Thank you so much, Eric. It was really lovely having you here.
Thanks for your great questions.
Maybe on another topic. Thank you so much for listening. If you want to be up to date with all our episodes, subscribe to our podcast, and I'll hopefully hear you one more time. Thank you.
Goodbye.
Thank you for listening. This was Where Technology Meets Science by Appsilon. If you know someone who might enjoy this episode, share it with them. And subscribe to never miss a new episode. At Appsilon, we empower pharmaceutical and life science companies to leverage open source technology for faster data-driven decision-making in regulated environments. Until the next time.